Okay, here we are in lesson 3, nitrogenase. Where does the dinitrogen bind to the cluster? How does it get there? How does the ammonia leave? So we understand a fairly good amount now about the mechanism of dinitrogen reduction from the previous experiments in the previous lecture, but we wanted to know more. We wanted to dive in and understand exactly the pathway by which the nitrogen goes in. We've been studying this enzyme for 30 years, and it would really be great if we could understand everything about it. So that's what this lesson is going to discuss, is how we could figure out this information and what that information is. Here's just a reminder of the big picture and the structural details of the nitrogenase cluster, the iron protein and the iron molybdenum protein. So these two proteins come together, and the ATP binding causes a electron transfer event to take place. So the iron cluster binds to the iron molybdenum cluster. Then it binds ATP, which causes a conformational change. ATP is cleaved, causing a subsequent conformational change, which prevents back transfer of the electron. The electron during this process leaves the iron protein and enters into the iron molybdenum cofactor via the P cluster. This process then has to repeat 8 times using 16 ATPs, 8 protons, 8 electrons. There's a reductive elimination of dihydrogen to bind and reduce the N2 at iron 6, presumably. I should note that at this time, it is known that carbon monoxide binds to iron 6 from other studies. Also, the process is reversible. In the presence of dihydrogen, this process can go backwards. The dinitrogen can be released and at the Janus intermediate. In the absence of bound N2, we can still deliver electrons and protons to the cluster, but H2 is lost in the reductive elimination step, and 4 electrons and 8 ATPs are wasted as the Janus intermediate goes back to state 0. So how does N2 get into the cluster? A lot of work was done to study this protein and to look for channels or pathways that nitrogen could enter and ammonia could leave. Lots of studies indicated several pathways. These were done by computations on X-ray diffraction data or molecular dynamics calculations. A water channel, shown here, is known to exist, and that was believed to be the source of protons that need to get delivered to the cluster, but dinitrogen may prefer a non-polar channel instead of a polar channel. An ammonia egress pathway was identified, and several hydrophobic channels were identified. So dinitrogen is hydrophobic, and so we would expect that it might go through a hydrophobic channel. Xenon is monatomic, but also neutral, polarizable, water-soluble, does not form hydrogen bonds. Its atomic radius is very similar to the NN bond distance, and it's easily detected by X-ray diffraction. Xenon NMR has shown that xenon binds to proteins, usually filling an existing cavity by displacing water or filling an otherwise empty pocket, and these tend to have little distortion of the protein. It binds using non-covalent van der Waals forces, so xenon tends to travel in the same places that we might expect nitrogen to travel through this protein. So, nitrogenase crystals were grown under an argon-hydrogen atmosphere from distilled water containing polyethylene glycol, lithium citrate, sodium dithionate, which is a reducing agent. It's a pretty standard type of crystallization technique. Then, these crystals were lifted through mineral oil, and that mineral oil serves as a cryoprotectant in a later step. So you're coating this droplet, this crystal, with mineral oil. And then, this is the cool part, they pressurized these crystals under a xenon atmosphere at 14 atmospheres for about 10 to 15 minutes before then being flash frozen in liquid nitrogen. So that's why you need that cryoprotectant. When they solved the crystal structure of 2 different proteins that had been treated in this way, the CP protein and the AV protein, they both exhibited multiple xenon binding sites, shown here. Other data showed from other crystal structures showed small molecule binding sites. IMD and EDO are imidazole and ethane diol. So those binding sites are present in this structure, as well as carbon monoxide, sulfur, and iron atoms present in this structure. So the location of the xenon is what we're going to focus on next, but these xenons had to get into this protein somehow, and so they looked at those xenon binding sites and tried to find pathways that the xenon could have followed to get to that location inside the protein. So here are some of the pathways that were calculated for xenon 1 and xenon 2. There's a lot of amino acids that are identified in this diagram. Those are all of the amino acids that the xenon would have to travel by in order to get to the iron molybdenum cofactor shown in the lower left here. You'll note most of these amino acid side chains are non-polar. There are some polar ones mixed in there. And the first channel starts at this end of the protein and can travel through to this location. So here's a xenon atom located in the diffraction. Here's another one. This second xenon atom probably has to come in this way. This one could have also come in from this channel. And then there's a pocket that leads to this face, which is the 2637 face of the cofactor, which was implicated as the correct face for the dinitrogen activation. Iron 6 here is the one that binds to carbon monoxide in other studies, and that's the one where the reductive elimination of the two hydrogen atoms was proposed to be taking place on this atom, although now we believe it's taking place by the bridging hydrides between these two atoms and these two atoms. The point of this is that they did locate a channel, and it does point to the 2367 face of the iron molybdenum cofactor. Here are some additional channels that are following the IMD and EDO, the small organic molecules that go into the structure. And these again start at the surface and lead to the iron molybdenum cofactor here. A variety of possible pathways here. These pathways lead to either the 2367 face or the 3457 face. I haven't erased all of the different amino acids that are listed in this diagram, but again, these are the amino acids that the substrate would have to pass by on its way to get to the cofactor. So there's multiple possible entry points into the protein for nitrogen to bind to, and there's also multiple possible exit points for the ammonia. The ammonia, being a polar molecule, could easily follow the hydrophilic channel, the water channel, but it might also go out through a different binding pocket. These are still unanswered questions, but there's certainly lots of pathways available. However, all of the data points to the location of the dinitrogen binding in the iron molybdenum cofactor on this 2367 face. One experiment that was done to sort of verify this is this valine 70, which was the only amino acid that was shown 2 slides ago. So you might want to go back and check 2 slides ago. That amino acid was valine 70, and it's sitting right in that pathway. It's blocking the top of this face. So using site-directed mutagenesis, this valine was substituted with 2 smaller hydrophobic amino acids, either alanine or glycine, and this then removes this isopropyl group, or removes all of the carbons, and it gives a much more open channel. And when this was done, the native nitrogenase enzyme the modified nitrogenase enzyme could reduce larger substrates such as propargyl alcohol. And so that suggests that the channel that terminates near valine 70 is pretty important for nitrogen activation. The propargyl alcohol can't fit when there's a valine here, but it does fit and go to the face when it has more space. The product of the reduction of that alcohol was detected bound to the iron atom, uniron atom, presumably iron 6. So that really suggests that this is the pathway that's really important for catalysis. So the mechanism of nitrogenase is pretty well understood now. The proposed mechanism is in agreement with all the experimental evidence, but it took a long time, 3 decades, multiple crystal structures, a central carbon atom. That central carbon atom is invoked to hold the iron centers together in a rigid platform that allows all of this chemistry to take place without a lot of molecular motion. Those hydrogen additions, nitrogen addition, reductive elimination all take place on a stable surface. And people are affectionately calling this nitrogenase enzyme as having a heart of steel, referring to the carbon and molybdenum additions to the iron atom. So molybdenum steel, carbon steel makes it stronger, and that may be a bit of a anthropomorphization of the cluster, but it certainly helps rigidify the structure. But think of all the techniques that people had to throw at this problem. X-ray fluorescence, XAFs, EPR, eSeam, X-ray crystallography, high-pressure X-ray crystallography. Bioinorganic chemistry is extraordinarily difficult to study. These proteins do not lend themselves to easy study. And you have to throw a lot of different techniques at these problems. So what does the future hold for nitrogenase? Is it going to be possible to synthesize small molecule analogs that have nitrogen splitting activity? Can we incorporate this protein into other plants or other bacteria that don't have a symbiotic relationship? So could we put this protein into a plant and have the plant grow its own fertilizer? We would be able to then control the nitrogen cycle without relying on bacteria for more than half of our fixed nitrogen on the planet. So our understanding of nitrogenase is still a work in progress, at least in terms of its application towards utility for the world. The last lecture this week is going to talk more about the philosophy of science and how nitrogenase guided my career and the careers of many other people over the last 25 to 30 years. So I'll see you there.